Good morning, everyone. As said, the topic given to me is sterilization and sterilant. I will be following. I'm sorry, I can't share my screen because my system is faulty.
Go ahead, go ahead.
So I have an outline.
Can you show your video? Let's see your face.
Okay.
Yeah. Okay, you can go on now.
Right. So, introduction. Sterilization is an important process that is being designed to eliminate all forms of microbial life. And as we know, we have various types of them. We have bacteria, we have fungus, we have viruses, we have the spores. And based on all of them are present everywhere. So it make it to be the sterilization to be a quieter process in most of the field, especially when we talk of healthcare, in pharmaceutical company, food processing, as well as laboratory settings in every research. So even a single microorganism can lead to a serious consequence, as we know, and that is why there is need for sterilization. And in the case of sterilants, they are chemical agents that are specifically designed to achieve also sterilization, the same aim of sterilization. But in the case of stelulants, they are either in liquid form or gaseous form, and they are being used for transitional heat sterilization as well. Then we have various methods of this sterilization. We have physical method, then we have chemical method. The physical method involves sterilization by heat. It can either be moist heat by applying moisture, or by dry heat. When we talk of moist heat, we make use of instruments like autoclave, whereby whatever we are sterilizing is going to be subjected to a certain degree of temperature, which is 121 degree, for between 15 and 20 minutes under steaming with that particular pressure. So it is very, very effective in killing most microorganisms. Then we also have dry heat. Based on the material we want to sterilize, the dry heat is always being operated at the temperature of 160 to 180 degrees, and for longer periods of about an hour, for just to terminate the existence of this organism. Then we also have radiation process, and we can either have ionizing radiation or non-ionizing radiation. When we talk of ionizing radiation, we are referring to use of gamma rays or X-ray. These ones are able to penetrate material, and they are able to destroy the DNA of microbes. Then we have non-ionizing. In their own case, it involves the use of ultraviolet rays. Just as we at times use in the lab, we use ultraviolet light, and it is very effective in sterilizing surfaces that have limited penetration. Then we have the chemical method of sterilization. This involves the use of gaseous sterilant as well as the liquid sterilant. In the case of gaseous sterilant, we have ethylene oxide, which is known as ETO, and this one is very effective for materials that are heat sensitive, and it has a wide spectrum. That is, it can terminate or wipe off many microbes. Then we have ozone. The ozone one, that is inside and under the gaseous sterilant, they are also a strong oxidizing agent, They are used in sterilizing the air, that is the environment in which our laboratory experiment is being carried out or research. So it can sterilize the air, surfaces as well as water. Then we have liquid chemical sterilant. This involves the use of glutaraldehyde, and it is very common in medical apartment. That is whereby they use it in sterilizing their equipment, and it is very, very effective, but it only require careful handling because they are very toxic. Then we have peracetic acid. In its own case too, they are very, very effective, and it is rapid in its function. So they are used in food processing company and also in medical establishment to sterilize materials. Then we also have filtration. This filtration, we have a membrane filtration, that is using filter that has four sizes that are in micro. These are they are very tiny to retain bacteria as well as other larger microorganisms from the liquid or gas. Then there are also some other common sterilants. We have ethylene oxide. Ethylene oxide is a colorless gas, and they are belonging to the group of alkylate. Then we have glutaraldehyde. It is in liquid form as well, and it requires exposure for long period. We have hydrogen peroxide, which is also very active against pathogens, and they are commonly used in their gaseous form. Then we also have peracetic acid. They are known to be strong oxidizer. They penetrate cell membrane and disrupt the cell membrane of a particular microbe. Then can we apply these various methods which I've made mention along the way? We can apply them. They are useful in healthcare. They are useful in pharmaceutical company, that is where drugs are being produced. Then also in food processing, and also in various laboratories where research has been conducted. Then there are some things that need to be considered while we are applying this sterilization method, because they are used based on the type of materials that we want to sterilize. Without consideration of materials, there may be error in the application of this sterilant. So there are some consideration and best practices that needs to be taken into consideration. Like there is need to validate and monitor whatever the technique we are using. This is by using, especially when the sterilization process are being carried out, they need to be validated, and there must be routine monitoring. This is by using various biological indicators to ensure how effective most of these sterilants are.
Dr. Tomilayo.
So as to, yeah.
Yes, I need to stop you here. I don't want us to take more than 10 minutes. So we'll use the
Okay.
remaining time. You're already 9 minutes now. So we'll use the remaining time to ask questions. So, but next time you are going to take us more on this subject matter.
That's even the last slide.
Yeah, I know. Let's use question to take the last time. So if you have any question, please ask your question now. If you have any question on what has been discussed, you can ask your question. Okay, I have a question, ma. My question is that my question is you put a material in the autoclave. When you are removing it, how do you know if it has been autoclaved or not? Because autoclave is supposed to sterilize it. How do we know if it is sterile or not?
It has a particular temperature and the particular number of minutes. So to know whether it is being autoclaved, you have to make sure that before beginning to time, you have to make sure that the autoclave has warmed up and has started put on autoclave with when it is subjected to pressure and heat.
All right.
So
Thank you. Thank you, Dr. Tomilayo. Thank you, ma. I will end the Zoom and then I will start it again because I have 40 minutes on the Zoom. So the next speaker will, as soon as we come in, the next speaker will start speaking. Please. Thank you.